Hello everyone, Mark Skolnick here. I'm in the hills outside Parma, Italy, where I spend half the year now that I'm retired. In the calm here, I'm able to relax and make a video with Jerry Launchbury coming in remotely from Salt Lake City. Our thoughts have turned to genome-wide association studies, or GWAS, and how the disease-associated SNPs are perfect starting points for gene discovery. Why haven't they been used before for gene discovery? Two reasons come to mind. The first is that high-quality full genome sequencing of the samples used in a GWAS has been out of the reach financially for many researchers. That's why chips with 1 million SNPs were used. But now that one can explore the full 3 billion bases of these samples at a reasonable cost, a new discovery opportunity arises. Before explaining what can be done and what can be found, we are going to review some basic principles. Jerry has been focused on modern genetic diagnostic testing for common diseases. These two seemingly disparate topics actually share a lot in common, so we decided to make a video that links them. I was the scientific founder of Myriad Genetics and its first chief scientific officer. When Jerry was hired, we ran research together, and when I retired in 2010, he became the new chief scientific officer until 2022, when he also retired. Our discussions have been fundamental to the creation of this video. After that long-winded introduction, let's take a look at some slides. We will try to convince you that your GWAS data and samples put you in an amazing position for gene discovery, improvement of polygenic risk scores, and understanding of gene regulation. We then turn to the present and future state of genetic testing and describe the progress that has been made in genetic testing with a case study of breast cancer genetic predisposition. There are thousands of GWAS data sets, and each one has the potential to discover genes for a predisposing disease or for a trait, depending on the study. This is possible today, just as it was not possible in the past, due to the dramatic drop in the cost of full genome sequencing. Since low-cost sequencing amounts to a paradigm shift, we are making this video to alert all of you, not just those of you at large sequencing centers, that the game is afoot and that the fleetest will make great discoveries. Why are we not doing this ourselves? We're both retired and we don't have a laboratory now. So all we can do is pitch the ball to you and wish you the best of luck. A second purpose of this video is to alert you to the great advances that have been made and are being made in genetic testing, and to advise you to prepare for a new genetic diagnostic paradigm.
Hello, I'm Jerry Launchbury. Today's sequencing costs are so low that there is no longer a barrier to gene discovery or to genetic diagnosis. Many of you have access to sequencing through your universities or their commercial associations. For those of you who don't, several commercial services exist which are able to provide you with high-quality 30x full genome sequencing at a very reasonable price, say about $450 US. Certain services will also return polygenic risk scores for common diseases and traits. For those of you who have IRB permission to return a full genome sequence and disease risk analysis to subjects who provided you with samples, you're in a great position to assist both cases and controls in their healthcare. What we're going to describe is well known to all GWAS researchers and many other viewers. But for those not in the field, we are going to explain the background science as well as the discovery approach. We will also give enough supportive material so that viewers not in the field can have a good understanding. What are polymorphisms? The normal human genome contains 3 billion bases of DNA. Each of these A, T, C, or G bases in a normal sequence could have any one of the other bases as an alternative, inherited from the other parent, and this could predispose to disease. These alternatives are called polymorphisms when relatively frequent. This diversity exists at both the species and the population level. Given the amount of DNA sequencing that's been carried out in various human populations, we now know that around 20 million of these bases differ in humans. Is this an upper boundary? Surely not. Assuming each living member of our species has one unique DNA change, then sequencing 3 billion humans would provide an example of almost every changed base. We could theoretically interrogate this diversity for its effect on predisposition to any human common disease or trait. In practice, with the exception of a few biobanks, only about 1 million of these differences have been examined in humans so far at the research and consumer level. Several thousand polymorphisms have been shown to have detectable effects on common disease development. The individual effects are relatively small, and for convenience, these effects have been combined into polygenic risk scores, or PRSs, for each disease studied.
To study the risk effects of these polymorphisms, genome-wide association studies are performed with a chip that reveals polymorphisms at 1 million or so loci. Each locus on the chip contains 2 sequences, one for each allele. Each chip is hybridized with the DNA from a case or a control, creating a data set of the genotypes of each of the SNP loci. In this hypothetical example, we see an individual who has a G on the chromosome from one parent and an A on the chromosome of the other parent. This genome-wide association study shows that 20 loci showed an increased risk for kidney stones when cases were compared to controls. The x-axis denotes the chromosome position. Note that the p-value for significance on the y-axis is less than 1 in 10 million, since a large number of tests were made. The higher the association, the bigger will be the risk for disease caused by that locus.
Many tens of thousands of these single nucleotide polymorphisms, or SNPs, have been shown to have detectable effects on thousands of diseases and traits. The individual effects of SNPs are usually small, so they are combined into what is called a polygenic risk score, or PRS, for that disease or trait. PRSs are proficient at separating individuals into risk categories. People who have high polygenic risk scores can have lifetime risks for a disease equivalent to carriers of intermediate risk gene mutations. People with low polygenic risk scores will have lifetime disease risks well below the average. One disadvantage of first-generation polygenic risk scores is they showed limited portability between populations of different ancestry. For some diseases, polygenic risk scores have been developed which may be used across different ancestral groups and in people of mixed ancestry.
One can view the differences in a polygenic risk score either as a curve or as an age-specific risk. In the first figure, we can see that the distribution of the risk in cases is shifted to the right from the controls. Age-specific risks show the cumulative risk of the high-risk group diverging from the medium and low-risk groups with increasing age. Now, finally, for the meat of the first part of this talk. When we talk about the specific sequence along a single DNA strand, we refer to it as a haplotype. In the GATC sequence, allele G is present in 30% of cases and only 20% of controls, demonstrating a 50% increased risk for individuals with a G allele. The second allele, an A, is present in 70% of cases and 80% of controls. Returning to the G or A polymorphism presented earlier, we now look down the chromosome of the G allele and we see another polymorphism, either a T or a C, and we can study the haplotype containing both SNPs. In our case, the disease-associated SNP divides into 2 haplotypes, either GT or GC. G was found in 30% of cases, which breaks down to 15% GC and 15% GT. However, the 20% G found in controls is divided into 15% GC, the same as the cases, and therefore only 5% GT. The increased risk associated with this SNP haplotype changes dramatically from 50% based on the GWAS associated SNP to 300% based on the associated haplotype. We can attest to the fact that the most excitement you can have in genetic research is in finding the gene you have been looking for. The suggested research in this video is to scan the sequence around the GWAS-associated SNPs with the whole genome sequence for each sample. This allows you to study all of the associations you have found for your disease of interest. For SNPs found in coding regions, further study is needed to determine between a causal SNP or simply another SNP in the region. The allele frequencies in cases and controls should give you a good indication. Then biological research may be needed to confirm causality. SNPs in non-coding regions would be more informative than the GWAS SNP if the frequencies in cases and controls show improved discrimination. If the sequence around the new SNP is recurrent across numerous SNP regions in your study and others, then you may be developing knowledge of regulatory regions. Most insertions or deletions in the coding sequence will completely disrupt the codons produced, and the gene will be dysfunctional. Insertions or deletions of 3 bases or a multiple of 3 won't disrupt the codon structure, but will add 1 or more amino acids to the protein the gene codes for. It only takes one case with a mutation like this to be able to say that you have found a causal gene. A larger sample, still small, would give you a better chance of finding it. If you sequence a single case and it has this type of sequence change, then you have discovered the deleterious gene that created the increased risk seen from the GWAS SNP. There are lots of other ways to destroy a coding sequence. If the start codon is altered or if a stop codon is created, then the normal protein will not be created. Similarly, a splice site mutation will destroy splicing a pair of exons together. Also, there are tandem repeat mutations which can alter the number of repeats, resulting in no protein or an abnormal protein produced. As above, just a single sample's abnormal sequence is enough to determine that a causal gene has been found.
Of course, not all the predisposing genes will be found, as this gene discovery approach is based on the original finding of linkage disequilibrium between a SNP and causal gene variation. Linkage disequilibrium will not always occur, as there could be multiple haplotypes that carry the causal gene and no association may be detectable. Similarly, if there are many different causal mutations in the gene, there won't be an association. SNP chip content is selected from polymorphisms with relatively high frequency. Associated with rarer sequence variants may be missed. SNPs on most chips occur on average every 3,000 bases of DNA, so many genes may not be in a covered region. We presume that whole genome discoveries will open up pathways for further genetic and biochemical discoveries. Gene-gene and gene-environment interactions will also be discovered and will enrich our understanding. Let us now turn our attention to genetic testing for breast cancer risk. We've chosen to showcase breast cancer as it is the best example of the integration of traditional monogenic risk and polygenic risk along with other factors. This approach has been introduced into women's healthcare and represents a powerful new paradigm in preventive medicine. Our best estimates of the genetic contribution to breast cancer development show that around 35% of the variability in risk is due to inheritance. Of this 35%, about half has been accounted for by mutations in a combination of high and intermediate risk genes such as BRCA1, BRCA2, PALB2, CHEK2, ATM, and others, and by polygenic risk scores. Discovery of genes for associated SNPs and other approaches are needed to discover more of the missing half of heritability. For women who are not carriers of mutations in any of the 12 known risk factor genes, risk is best evaluated using a PRS-based model combined with other factors. Useful models include Tyrer-Cuzick, Boadicea, and Gale models. These combined models are of particular clinical value for women who have significant family histories of breast cancer, but additionally have utility for all women. The evolution of our understanding of the genetic variation behind breast cancer development has been one of discovering strong individual effects first, since these were the standouts. The relatively rare individual mutations in BRCA1 and BRCA2 are the best examples of this class. Collectively, these mutations are found in about 1 in 200 US individuals, with specific mutations even rarer in the general population. In some special populations, such as Ashkenazi Jews or Mexican Latins, there are individual mutations found at relatively high frequencies due to the vagaries of population history. It's very important to note that these mutations are found in both women and men. Fathers can pass them on to daughters and sons. In total, there are around 12 known genes that are mutated and cause higher risks for breast cancer. Some of these are relatively rarely mutated, such as PTEN and TP53, and cause complex cancer syndromes. Others, such as PALB2, are uncommon and have risks approaching BRCA2. Still others, such as CHEK2 and ATM, carry relatively common mutations and encode intermediate risk for breast cancer. It took longer to attribute breast cancer risk to several of these genes due to rarity in the case of PALB2 and to weaker effects on breast cancer development for CHEK2 and ATM. Let's talk about penetrance. Variable penetrance occurs where, despite inheriting a disease-predisposing mutation, the subject does not develop the disease over their lifetime. One strong factor driving penetrance variability is the cumulative effect of numerous individual DNA risk factors across the genome, the polygenes. As we've discussed earlier, the cumulative effect of polygenes can be captured in a polygenic risk score. A bad hand of polygenes raises the probability of developing breast cancer in a carrier, while a protective set will lower the risk. This is likely to be true for all common diseases in which genetics plays a role. To identify women at risk of developing breast cancer, it is essential to combine the effects of high-risk genes, intermediate-risk genes, and polygenes with all the other known clinical, biological, and lifestyle risk factors into a complete model. The 12 high and intermediate risk genes should be fully sequenced. Ideally, major gene risks should be modified by ancestry-appropriate polygenic risk scores. This has been achieved for women of European ancestry for CHEK2 and ATM, but data for women of other ancestries are lacking. For women who are not carriers of mutations in any of the 12 known risk genes, risk is best evaluated using a polygenic risk score model combined with the classical Tyrer-Cuzick, Boadicea, or Gale model. These models are of particular clinical value for women who have significant family histories of breast cancer, but they have utility for all women. While genetic-based models are helpful to most and critical for many, there are significant challenges that remain in implementing PRS clinically. Barriers to adoption include the need for ancestry-specific studies and the general lack of provider education and society guidelines. A recent study of which I was an author showed the power of biological models that incorporate all ancestry polygenic risk scores to predict the development of breast cancer. In this important example, the model including PRS better predicts breast cancer development over a 25-month period than a standard clinical tool which is in wide clinical use. You can see from the upper graph that the PRS-based tool is better in general at predicting risk compared to the standard Tyrer-Cuzick tool. What is crucial here is the lower panel. This shows that the PRS-inclusive model has the ability to predict high risk in women that the standard tool says are low risk, and for many women that the standard Tyrer-Cuzick tool says are high risk, the PRS-based tool correctly predicts their low-risk status. Similar approaches are taking shape in colon cancer, cardiovascular disease, and other conditions. And while the adoption of these approaches is both patchy and incomplete, the promise of such technology and the degree of enthusiasm surrounding its potential adoption sets us up for a phase in which the description of genetic risk is more complete through whole genome sequencing. Mark and I have gently reviewed the current state of the art in human genetics and its explanation of variation underlying complex disease and to some extent human traits. What is clear is due to a combination of pathogenic major gene effects and polygenic risk and modification factors combined with clinical, biological, and environmental factors. With our teams, both of us have been intimately involved in the discovery, validation, productization, and commercialization of important predisposition and companion diagnostic tests in breast and ovarian cancer over the last half-century. However, we are sanguine enough to realise that these models, while useful today, are not good enough long term. Rare and ultra-rare gene variants await discovery. Perhaps some or many of these will be private or almost family-specific. We need to understand the genetic functionality that exists under the surface of each known and validated set of polygenes. Part of the reason for making this video is our enthusiasm for the next phase of discovery in complex human disease, which keys off technological improvements in sequencing and severe cost reductions. Ultimately, in the next 20 years, the community will generate more powerful risk models for both the clinic and consumers as people take enhanced control of their healthcare options. These advancements will affect many aspects of people's lives and the legacy their DNA bequeaths to their families.
We live in a beautiful world and at a unique moment in time for healthcare. Previously, it has not been possible to examine a human genome sequence at such exquisite detail for less than $500. Our ability to mine vital healthcare information and to translate that into positive healthcare outcomes is an opportunity not to be wasted. I'm excited for the future and wish you the very best fortune as you race forward on that journey. Surely, this is the dawn of a new era. To make this video, I was fortunate enough to find a fantastic production team. First, there was the director and producer, Jacopo Gianni. Then the director of photography, Alessandro Fioroni. The camera operator, Isaac Bidioli. The other camera operator, Michele Ghisoni. The sound equipment operator, Federico Montaldo. And the teleprompter operator, Luca Tosetto. And myself, the alternate teleprompter operator.
Langsam.